So, mastering Western blotting, optimization, expert tips and techniques for better results. This presentation has been prepared on questions which I have received continuously about Western blotting, which is rather complex technique, as you probably noticed yourself. A little bit about where Agrisera is located. We are located up north, not at the polar circle yet, but 1 hour flight from Stockholm, capital of Sweden, Stockholm. And at the moment we are experiencing polar night, so the days are quite short. And in the summer we are having a constant day. So this is rather exciting place to live and to work. And a little bit about Agrisera, which has been established quite a long ago, 1985. Our farm is located in this beautiful landscape of northern Sweden. We also have a facility in Umeå where our antibodies are purified, QC tested by Western blot and ELISA, and our cataloged antibodies are delivered from Umeå. Agrisera has 55 employees and we are constantly growing. We are part of Olink from 2020. It's a company which is providing very exciting platform for quantification of human proteins, and Agrisera is developing antibodies for Olink technology. We are also ISO certified and have been working with antibodies for quite many years. And now we are focusing on Western blot technique. That technique, I don't know if you are aware how old it is. Think in your head, which year do you think this technique has come to be used in laboratories all over the world? It allows to detect specific protein in a complex mixture. So, look at the date of the first publication, September 1979. So you're using a technology which has been around for quite a while. And it has been independently established in 2 laboratories. You are probably familiar with other techniques, southern blot, northern blot for RNA, Eastern blot for post-translational modifications. And I wonder if you know why Western blot, where is the name coming from? It's not to fit the other names, it is actually a coincidence. The laboratory which was establishing this technique has been located on West Bank, therefore the name Western blot. And as you know, this technique is based upon a concept of a sandwich. I hope you had your lunch, and we are diving deeper into how the sandwich is built and how it's just a reminder for you of how Western blot technique is working. So we're starting with basics here. You separated your proteins on a gel of different percentage. And what we are after is to obtain an exact copy of everything what was on that gel on a membrane. And that is done by applying electrical current through so-called sandwich. So you're putting layers of, this is of course a wet transfer I'm showing here. So the layers of the Whatman, and then in between we have gel and a membrane. We are applying a current and proteins are migrating from the gel to the membrane. And we are after an exact copy of what we had on the gel to be transferred to a membrane. And who is doing a work with actually visualization of your target protein? It's an antibody. And here also a reminder, we're starting with the basics, how the antibody is looking. It's a Y-shaped molecule, and we have an FAB region where antigen binding domain is located. That's the most important part of the antibody. It's so-called variable region, and that's where the fit between antibody and your target protein is. And also we have so-called FC region of an antibody. It's a constant region called because when the research was done on this immunoglobulin, that fragment crystallized in a fridge, and therefore it's called FC region. And that's a constant region. It's not changing, and it has affinity to protein AAG and AG, and allows protein antibody purification. So the binding to your target protein is happening here. Of course, we also have shorter antibodies. Maybe you heard about them. It's antibodies from llamas. As you will see, they are smaller. And it means also easier penetration, especially interesting in techniques like immunohistochemistry. And what we are after is the binding. So we want the antibody to bind to our target protein. A little bit more of the basics. Antibodies in the antigen binding domain are binding to so-called epitopes. And as you see here, we are having a linear epitope, which consists of subsequent amino acids, in this case, 3. And the antibody is binding to this linear epitope in techniques where proteins are denatured. We're dealing with linear epitopes in Western blot and immunohistochemistry. So please keep this in mind. It's going to be important later. We are also having antibodies are also binding to so-called discontinuous epitopes, which are brought. Distant amino acids are brought by protein fold. And these epitopes are detected, or antibodies are binding to in techniques where native protein is recognized, like immunoprecipitation, Western blot in native conditions, or chromatin immunoprecipitation. So remember, in Western blot, we're mainly dealing with linear epitopes because proteins are denatured. You probably heard about the concept of poly and monoclonal. We are using poly or monoclonal recombinant antibodies. I'm bringing it up here because oftentimes what I see is that people consider monoclonal antibodies to be better. And yes, they are very good, but there is a very big difference between how these poly and monoclonal antibodies are working. And I am going to show you. So poly means many. Polyclonal antibody will detect a pool of different epitopes. So it can be 3 to 15 amino acid long epitopes, linear or conformational epitopes, which polyclonal antibodies are binding to. Okay, poly means many. Mono means one. So one epitope is only detected. An epitope can have 3 to 15 amino acids. So if we have a monoclonal antibody, it will bind to only one epitope. Of course, it may mean less background, but if such antibody stops working, it stops working for all and for good. And also, it may not be suitable to more techniques. It may work only in one technique. While polyclonal antibodies are binding to a pool of epitopes, this means that they may be more versatile and can be used in a range of different techniques. I will share the slides with you afterwards, so you will be able to have both slides and presentation. So remember, poly and mono, and it doesn't mean that one is better than the other. They will be suitable for different techniques depending what you're working with. To succeed in Western blot, we need to have antibody matching species and application. And that's often actually overlooked. Let me explain what I mean. When we design the peptides, we want to cover oftentimes as many species as possible to save on animals and on the cost. So we design antibodies to short synthetic peptides, and we call them so-called global antibodies because that sequence, up to 15 amino acids long, will be existing will be present in the target protein from multiple species. So this graph depicts it. So we have this antigen binding domain, this sequence. Let's say we have a polyclonal antibody which binds to 15 amino acids, and these 15 amino acids are repeated in the same target protein from these different species. How do I choose the right antibody for my protein? Here goes an example. Peptide antibodies are made to short sequences, and the sequence can be coming from N-terminal, from the middle of a protein, or from the C-terminal. Of course, the signal peptide in this case has to be not included when we are designing the peptide. And you need to keep in mind that that sequence antibody is made to need to be repeated in the protein you aim to detect. And this can be checked on a sequence level before you even go to do the blot. So this is often overlooked. We receive antibodies from the colleague, or we really want the antibody to work, and then we spend hours in the lab testing, while we could do this sequence analysis. And from the sequence analysis, we can obtain the knowledge if an antibody has so-called predicted reactivity. Therefore, on our website, you will find on product info sheets confirmed reactivity. It means that antibody is binding based on experimental data, and predicted reactivity, which means that the sequence the antibody is made to is found in the species which are listed. Of course, sometimes antibodies are made to proteins, and then by aligning your protein and the protein antibody was made to, you can obtain the data if there is enough of sequence identity. This is extremely important. And before you use any antibody, please make sure that this sequence conservation is occurring. Of course, you can say that this data to what antibodies are made is not available on the website. And that's true, but you can always ask the company you aim to purchase antibody from. So please make sure that, because people often align whole proteins, but if an antibody is made to the peptide which is not conserved, the antibody is not going to work. Short reminder how antibodies are binding, how we are making the visualization. We have a target protein on the membrane. Membrane is blocked. Then we are applying primary antibody, and to enhance the signal, we are applying a secondary antibody with a label, which can be an enzyme or a fluorophore. But it's also possible that primary antibody is directly labeled, especially for proteins of high and medium abundancy. This can shorten your Western blot protocol if you use directly conjugated antibodies. And of course, we have here a Western blot workflow, which contains quite many steps. I'm going to walk you through those steps and discuss different points and different questions which I have received over the years. So, of course, after the whole procedure is completed, we want to have our nice Western blot. Well, it's not always the case. The theory is good. How about the practice? Here, an example. The same primary antibody has been sent to four laboratories, and you can see the results. The sample is Arabidopsis thaliana. They were running different protocols, and they were aiming to detect PIF, which is a transcription factor, a protein which is not so easy to detect, and therefore a special growing conditions for the plants and extraction protocol have to be applied. And we have received these results. Our antibodies usually tested at least in 2 independent laboratories. So based on the first 3 results, one would say this antibody is garbage, it doesn't work. But in this case, performing the whole procedure in a certain way was important to obtain the result. So ask yourself, how much do you know about a protein which is to be detected by an antibody? A lot of work is done at the desk before you go to the lab. You can save yourself time and pain if you do a theoretical analysis. We want the antibody to bind to the target. How much do we know about biochemical properties of a protein? Is it soluble? Is it membrane? What is its expression level? Is it high abundancy or is it low expressed? Where is it localized? On an organ tissue level or on a cellular localization, organellar localization. We had experienced cases, for example, with antibodies to PIN, detecting PIN proteins, which are located at 2 millimeters of a root tip. So, of course, if you're grinding a whole root, you're diluting your target protein. Therefore, this theoretical analysis is extremely important. Amount of target protein in a tissue is the next thing to consider. But the question is, how do I know that? You can check it in different databases, which are actually available online. We are using PAX, Protein Abundance Database, which also have data for other species and plants. And if a protein has a level of around 5 ppm, then we consider it as difficult to detect using antibodies. There is also a very nice e-plant provided by University of Toronto, where we have gene expression level for different species, where we can at least see gene expression, where should I expect the most of my target protein. I'm aware that mRNA level can be high, protein level can be low. Anyhow, we need some clues about the protein we are aiming to detect. Next thing, which is very important, is sample quality. This has also sometimes is overlooked. We have been analyzing samples which were a few months old and saw gradual degradation of a target protein. Therefore, the rule of thumb is like this. If you work with protein of low expression, use freshly extracted material. Do not store your samples for several months. They can actually degrade in the freezer. So protein samples have to be of absolutely best quality. We have different extraction methods. I don't know if you are using a bead beater, which allows to extract simultaneously several samples, up to 24 samples. We need to choose the proper bead site, depending, especially for organism with rather robust cell wall like diatoms. This is a really good method to extract the protein, and it is fast. I have been working with time-consuming extraction methods, and I do not recommend them if you are processing several samples. There can actually be degradation during extraction. So, for example, protein pattern in sample number 1 will not be as in sample number 12 because certain proteins may be degraded. Therefore, if you have access to bead beater, I would like to encourage you to try this method. Protein extraction method for low abundancy proteins includes fractionation. I know that it is not a popular method, but if you are working with low abundancy protein, you are not going to detect it in total cell extract. I think quite many antibodies have been wasted because people got discouraged by testing total cell extract, no band, okay, the antibody is bad. But please check on this example here. By fractionation and by isolation of photosystem 1, the target has been concentrated, and therefore also, as you see here, the background pattern has changed. Some background has fallen off, and we have very nice detection of our target protein. So if you work with proteins of low expression, transcription factors, for example, I recommend fractionation. Fractionation doesn't necessarily mean that it is a laborious high-speed fractionation and protocol lasts for several hours. There are different methods to do it. Protein extraction method also involves certain buffer composition. This is a result from one of the test labs which have changed the buffer. Most commonly used, I know, is trees. I'm not so fond of it, but it's the most standard buffer. With more difficult proteins, better results may be obtained with, for example, hippies. And on a second example, if you wonder what was changed in this extraction buffer, we have barley Arabidopsis rice. The peptide is conserved in all 3 species, but on the first blot, the band comes only in Arabidopsis. So what has been changed? It is urea. So 6 molar urea has been included in extraction and loading buffers. What urea makes is that it unfolds the protein. Some proteins actually may partially refold during Western blot technique. So this refolding may hide the epitope the antibody is binding to. So by introducing a highly denaturating agent, one can keep the protein unfolded. Next thing, consider if we need to extract protein, we are aiming to detect from a specific organ. For example, from senescent leaf, from a 2 millimeter root tip. Of course, we want to include protease inhibitors. They are like a basic to be included, although in some cases they may induce protein modifications. So but this is like a deeper level of the Western blot. In general, protease inhibitors should be included, and also proteasome inhibitors if your protein is the target of proteasomal degradation. Of course, a proteasome inhibitor has to be included to extraction buffer. And we had a case of a lab. We developed the antibody. They have been using it for 1 year, and there was basically result was like this. They didn't want to give up. The project was important. And in the end, they decided to give it last try with proteasome inhibitor. What happened? There is a band because the target protein they were working with was exactly also a target for proteasomal degradation. And when you receive the presentation, you will be able also, if you want, to check following these links to check for more information. What things should we take care of during SDS page electrophoresis? I wonder if you know this, shortening GLP, good laboratory practice. What does it mean? It means that you're working clean. And I mean, really, really clean. Western blot equipment, all trays you are using for incubations, they need to be clinically clean. I was surprised to learn that not all labs are running their Western blots in the cold. So either in a cold room or in ice bucket with cold buffers. You need to be precise and you need to have fresh reagent stock. And of course, follow the protocol. So if it's written 5 milliliters, you are incubating in 5 milliliters and so on. So you really need to be very precise and consistent in your behavior with Western blot. I received a question about gel electrophoresis. We have different gels available. And here it's an example from Thermo Fisher. You see with naked eye that in this case for high molecular weight protein using tris acetate gave a little bit better bands, and the detection was also quite sensitive. I know that many of you is maybe casting your gels yourself. This is completely fine, but precast gels will produce reproducible and convenient, are convenient to use, and will enhance your reproducibility. We are using a gel system from Novex, where by changing the buffer, we can obtain different molecular weight separation of proteins, which is very convenient as we are working with proteins of very different molecular weight. So when we are using precast gels, we can play more with molecular weight range. They may have higher stability and longer shelf life. What is the difference between polyacrylamide gels? That was also one of the questions I received. Bistris and trisglycine. You do not need to remember this, but I just want to point out that depending what you're after, you see here, if you're working with, you want to minimize protein modification, Bistris gel is of choice. It is said to have sharper bands, but then trisglycine will give you a lower resolution for high molecular weight proteins. So if your target is of higher molecular weight, maybe you should consider, when you're refining your results, maybe you should consider another gel type. Trisglycine is also hydrolyzing over time, so shouldn't be stored for prolonged time. So everything depends upon what protein you're really aiming to detect, what molecular weight it has, and what downstream application you're using. What should I load per well? That was also one of the questions, and I know that what we recommend is microgram of protein or microgram of chlorophyll. But I was very surprised to learn that many labs are loading on volume. And the argumentation for this is that it's difficult, it's not possible to measure protein with denaturing reagents, with detergent, and therefore we're loading on volume. It is not recommended if you want to compare the samples, because you're not able to extract protein in a reproducible way if you're doing it by hand. And also such results will be difficult to compare. There are reagents which help to measure protein in actually extraction or loading buffers. So one can estimate protein concentration in presence of these different chemicals. So if you can, please consider loading on protein or on microgram of chlorophyll for photosynthetic tissues. How much material should I have? Is it enough to have nanogram of microgram? Of course, if you are separating and aiming to detect a recombinant protein, then it's enough with nanograms, because otherwise, if you use chemiluminescence, the whole membrane will be dark very fast. But otherwise, we load on a microgram of proteins. And here in these examples, you can see how we can titrate. Once we know how antibody is working, we can find protein load per well and dilution when we obtain very nice signal-to-noise ratio. In this example, you can see how the background became diluted. I mean, yeah, signal-to-noise ratio is improved when the primary antibody has been used in a higher dilution. Background signal has been diminished. Another question was, should I use 1D or 2D gels, and how this will affect the results? Of course, 1D gels, one-dimension electrophoresis gels, are most commonly used, but I would like to remind you that each of such bands will contain hundreds of proteins. So in the second dimension, the band is separated into specific proteins into different spots. And usage of 1D or 2D gels depends upon your questions and what you are looking into. So 2D proteins are suitable for proteomic, 2D gels are suitable for proteomic analysis when we want to detect hundreds or analyze hundreds of proteins. Sample denaturation. That was one of the questions which I received also before this workshop. Of course, there are all different variations which you see in literature and in different protocols. We are using 70 degrees for 5 minutes. However, for some proteins, another denaturing temperature is necessary. I do not recommend extensive boiling of your samples because it can lead to aggregation of proteins and lack of a band on a blot. A classical example is H plus ATPase, which is a plasma membrane protein. If a sample is treated with 90 or 100 degrees, H plus ATPase will aggregate and there will be absolutely no signal on the blot. So please consider in what temperature you are denaturing your samples. When it comes to protein transfer conditions, there's also certain things which should be considered. Here you can see how we are working. We are using ice buckets and cold buffers when we are doing gel electrophoresis and protein transfer. How important are the conditions of the transfer? Is it better to run semi-dry transfer in a cold room to avoid overheating the instrument? I think you already know the answer. I am a fan of cold rooms, and the Western blot procedure should be done in a cold. But here is an important thing depending upon what is molecular weight of your target protein. And this is also very often overlooked. We're taking one and the same protocol for high molecular weight and low molecular weight proteins. High molecular weight proteins will have it a little bit more harder to migrate from the gel to the membrane. Therefore, if we use membrane of 0.45 micrometer pore size, it's better for these proteins. They love SDS. And also it is said that wet tank transfer gives better results. Of course, there are exceptions. When we work with proteins below 20 kilodaltons, we should decrease SDS, increase methanol, and also use membranes of lower pore size. What happens in case of low molecular weight proteins is that oftentimes they are blasted through the membrane. So the reason why there is no band on a blot is because the protein is in a transfer buffer. So depending upon molecular weight of your target, you really need to consider your transfer conditions. High molecular weight proteins may require a transfer, for example, to be conducted for a longer time.
It's for maybe 1 hour more.
No, I think I will finish not, I will not take your 2 hours. I'm pretty efficient, but I think we will finish after 90 minutes. So if you have any questions, then you're welcome also to put them in the chat. Protein transfer type, there are two different types, and you probably work either with wet or with semi-dry. What are the differences? Amount of buffer. We use more buffer for wet transfer. We use less for semi-dry. Electrode contact is different. And speed, of course. Semi-dry is faster. Faster is not always meaning better. And I don't want to say that one is better than the other. It depends upon your protein. We are using wet transfer where there will always be exceptions. I've seen beautiful results obtained with semi-dry transfer. If a protocol is well optimized and suitable for protein antibody pair, then it's going to work very well. Which membrane to use? I can bet that most of you is using PVDF because I see that's a rather popular membrane and we are using it too, but there are certain differences. how proteins are binding, for example, to nitrocellulose. PVDF is more robust, can be air dried and rewetted. Nitrocellulose is more fragile. And here, I will not tell you that one is better than the other. My answer is it depends upon the protein. For some proteins, better effects, results are obtained with nitrocellulose, for others with PVDF. So actually, it would be good to have both in the lab and try on one or the other. There will always be exceptions. Air bubble removal is also an exciting step of Western blot procedure. Maybe you're doing it like that. Well, don't. I would suggest to do it like this. So put your sandwich in the cassette and then check for air bubbles. If air bubbles are on the top of the gel, we don't bother so much. What air bubble is doing is that there will be no correct protein transfer. And if it is located at your target protein molecular weight, of course, it will make it more difficult for your target to end up on a membrane. So exact copy of a gel is not going to be obtained. And also moving the sandwich can contribute to unsharp bands. So assemble your sandwich and then remove the air bubbles in the cassette. And also make sure, if you want sharp bands, that the sandwich is tight. So it actually, if it's too loose, you can add an extra pad to make it really compressed. Not too much, of course, but it shouldn't be too loose. I think you understand what I mean. We actually tested this recently. We have my colleagues, two Evelinas. One is more experienced, one is less experienced, and one was coming with quite sharp bands on her blot, and the other had unsharp bands. And I have asked them how they are doing their procedure, and this is how we discovered the difference between their protocols. Protein transfer quality should be somehow checked by, for example, Ponceau or Kumasi reversible staining. We're not doing it always, but if we are setting up a protocol, we will do that check. So of course, with a membrane which is looking like this, we will not proceed further with Western blot procedure. Protein transfer quality should be also checked by staining the gel with Kumasi. I'm not certain that you're doing it, but I would really like to warmly recommend it. And I would like to show you why. In this cassette, we had two membranes. And after the transfer, we stained with Kumasi gel 1, which was blank. This is how it should look, right? The protein migrated from the gel to the membrane. Although on a second gel, we seen quite a lot of protein still left. So this is how your gel should not look after Western blot procedure is completed. This means that you have maybe applied too little current, that it should be, or the transfer should be done longer. For when we work with protein quantification, we are not using this double sandwich. We are only using always one sandwich in the cassette. So remember, you can check if your protein left the gel by staining it with Kumasi, because there should be no bands, of course. Primary antibody format is also important for your success. And of course, you're familiar, probably you are using antibodies either as serum or purified. And purified antibodies can be to total immunoglobulin fraction, or if we are using antigen for purification, then antibodies will be purified to specific antibody fraction, which is just against the target protein. Please keep an eye also on product information sheets, because sometimes it looks like you're purchasing a lot of antibody, But if it is a total immunoglobulin fraction, then it may not be so much of your specific antibody in it, as you will see on this example. Because in total immunoglobulin fraction, only 0.5 to 5% is your specific antibody, because that purification is purifying all antibodies. But why we are using the antigen for purification, then we are pulling out our specific antibodies against the target protein. We are often getting questions how much of specific antibody is in serum. Actually, we don't know unless the antibody is purified, then we can estimate it. Purification is done on using chromatography on columns with specific matrices. So if antigen used to elicit a given antibody was a protein, we are using NHS activated Cepheros. And if it was a peptide, we are using Ultralink. So first we are preparing the column. We are coupling the antigen to it. Then we are applying a serum. As you see, it's a mixture of all possible antibodies. We're passing the serum through the column. The antibody to the target gets stuck, hopefully, and then we are eluting it by applying low pH, which makes conformational change on the antigen, and therefore the antibody is released and collected. Of course, this can also be conducted. We are using chromatography system, but it can also be done as a bench, lab bench purification, or in a mini scale on a membrane. Maybe you have been reusing the antibodies. This is also a very exciting question. Antibodies are quite expensive reagents, and I know that many labs are using Agrisera antibodies up to 15 times the same primary antibody solution. And how long can such aliquot be used? It really depends. For some antibodies, only 1 time, for others, 20 times. So this has to be empirically checked. And if you're disappointed that there is no signal, if you reuse the antibody, it means that all primary antibody bound to the target in the first incubation. There's nothing left to give you a signal when you are reusing that antibody. Antibodies of high titer can be reused. And of course, if you are working with uncharacterized antibody, please do not reuse it. You can do it with the antibodies where you know how they are performing. But if you are testing the antibody, if you haven't used it before, I wouldn't recommend to reuse it. This has maybe also happened to some of you. Primary antibody suddenly does not work any longer. What can be the reason? If you remember a concept of poly and monoclonal antibody, if we have monoclonal antibody, which is recognizing only one epitope, and that protein is losing activity, the whole antibody will lose its activity. There will be no signal. Polyclonal antibodies may lose activity gradually. So you may observe a gradual decline in efficiency of detection. And what are the reasons? The reasons are different. Sometimes improper storage, sometimes lack of stabilizers, and also using same antibody multiple times can also result in that you will not get a signal in the end. What is influencing antibody stability, if you remember the antibody binding domain, it's a part which is always different. And that one is influencing the stability of whole molecule, and it's no way to predict the stability of a given antibody. We had antibodies produced which lost activity after 1 year, and we also have antibodies which are active for 40 years. There is no software at the moment to predict it. It simply has to be empirically tested. I have received several questions here. and I'm going to address them. First question. Do you have any experience with turbo blot system and membrane proteins? Well, what I see is that it really depends what protein you're working with. So for some proteins, I've seen very nice results and not so nice in other cases. All this is a combination of uh, protein biochemical properties, uh, what protocol you're using. So I think you have to, if you have access to this equipment, you have to simply give it a try and see. With Western blot procedure, one has to be open-minded. There is no protocol will fit all protein antibody pair. My question is, how can we predict a good reactivity of an epitope in the sequence? Do you have programs of prediction to design a peptide epitope? Yes. There are specific programs for it. However, when we are designing our peptides, we have very experienced peptide chemist team. And we, of course, get support from algorithms, but it's always the experience of a human which is deciding if a given sequence is going to be used or not. There are certain rules here also, which sequences cannot be used. Sometimes peptides may be hydrophobic, not enough immunogenic, because depending upon amino acids, which are in the linear sequence, the peptide also may have solubility problems or antigenicity, having too low antigenicity. So yes, there are such programs which can help you. Do you have ideas to improve nuclear envelope protein extraction? Yes, I have some ideas, and on that question, I will answer by email. Next one, I usually stain my gel after transfer and oftentimes have residual bands even though the transfer occurred as the whole leather has been transferred and proteins also, as I can see them on Ponceau colored membrane. Should I still increase the transfer time? Or is it possible that I just have too much loaded protein at the beginning? Yes, it can be that you have too much protein. Remember, in Western blot, we are after signal to noise ratio. So, of course, if we are working with proteins of high expression like Rubisco, we don't need to load 30 microgram per well. So we can play with protein load per well. That's why I want you to load per protein, not per volume. We can play with the protein load depending upon what protein we are working with. For proteins of low expression, like Argonauts, I know that some labs are using so high loads as 100 microgram per well and more. And such concentrated sample can be obtained by TCA acetone precipitation, for example. So remember, we are after a good signal to noise ratio. So you have to sit at the desk first and consider what is your target protein? Is high abundancy, low abundancy? And depending upon that, you will choose your protocol. What are the sources of non-specific bands? Non-specific bands on Western blot we are always blaming antibody, but it can actually be that non-specific bands on the blot can be a result of that we have, for example, too much protein loaded, we are blocking too short, or blocking reagent is not concentrated enough. And this actually leads to a second part of my presentation where I'm going to discuss it. In a Western blot, also in publications, sometimes you see more than one band, usually at higher kilodalton. How can you explain this if the antibody is supposed to be specific? Well, I would say that the protocol is not optimized enough. And also, when we are developing antibodies, we always have to check them, validate them. So how we validate the antibodies? We need to use deletion mutants to be able to confirm that the antibody is binding to the right target protein. Of course, not in all instances we can obtain deletion mutants, and there are other methods to validate the antibodies in such case. There are certain instances where signal to noise ratio is not good enough, and therefore we will also obtain other background bands. And also you have to keep in mind that if antibody is binding to only 3 amino acids, 3 to 15, so it can bind to sequence of 3, 5, 10, 15 amino acids long, that these amino acids can be also found in other proteins, right? So sometimes the source of unspecific background can be due to that. I hope this answers your question, Anne. And now I will jump back to the slides and we will continue. And I promise that it will not be 1 more hour of the workshop, but think about your questions, which you will be able to ask me again after the second part. So hopefully you wanted to participate in a quiz, and I hope you found it fun. And now we're jumping into a very interesting issue, which actually is also connected to the questions which came. The polyclonal antibody is not good. It does weakly recognize my mitochondrial protein, but it recognizes much better many others. Even using purified mitochondria as a sample, can I poison this antibody by adding a protein extract from KO mutant to it? And is there a thumb rule for how much of this extract should I add? You see, people have problems. This is a real problem. It can actually be solved. Antibody background signal removal by depletion, it's called. So what you're gonna do is that you need to prepare a membrane where you are separating protein extracts from a mutant lacking your target protein. And of course, there will be all other proteins on it, but not your target. And then you incubate the antibody, which gives you problems, as you would do a regular blot. Okay? It means that these background antibodies will bind, hopefully, to the proteins. But since you do not have a target on your membrane, the antibodies to your target will remain in that solution. And then you can use them on the next blot, real blot. And that way, for example, if you have problems with cross-reactivity against Rubisco, from Rubisco protein, you can that way remove the antibodies which give you this cross-reactivity binding to Rubisco. This is a very neat method. And it may work, it may help you to clean your already antigen purified antibody or serum, which is giving you a problem. It is described in more details in a blog post. Membrane blocking and background. Now it is getting exciting because that's also an issue which is coming back, the background. Is blocking temperature sensitive? Should it be done at 4 degrees, room temperature, or in different temperature? It depends upon protein-antibody pair. I know that you heard it, but it is like that. And I'm going to show you why. Just a short note here. It has happened in our lab last week that my younger colleague blocked, and then she realized that she doesn't have the antibody to run on the blot. Panic, what to do? Have I lost the whole experiment? The answer is no. If it is PVDF membrane, it can be just dried. And she dried it up, protein side up, remember, left it to dry in the air, stored it, and used it next week with a very good result. And this is what actually, and please remember, don't use the tweezers as we are doing. They should have flat ends, not to destroy the membrane. And this is actually what we're doing. We are preparing a collection of PVDF membranes with different extracts on it, and then we are storing them for later use. If you have precious samples, it's actually better to store them on a membrane after transfer than in a freezer as an extract. And what happens also when the membrane is air dried is the protein will follow the nature. It will unfold. So if there are any issues with refolding and making that way the epitope inaccessible for the protein, if you air dry the PVDF membrane, then the protein will further be fixed further to the membrane. It's a very simple step in the procedure. We are storing such membranes up to 6 months. Membrane blocking and background, very exciting thing. We are using 5 to 10% non-fat milk, and this is what I see for protein, work with protein extracts from plant and algae. Usually the milk is giving a very good result. Of course, we have commercial blockers of proprietary formulation. Here, if you ever need a non-protein blocker, you can use PVP40. We also have BSA, but I've seen much worse results with BSA on plant samples. It's a small protein, and in non-fat milk, we have a mixture of a lot of proteins. Some labs are mixing BSA with non-fat milk. I don't consider it so necessary. And also we have gelatin, which is a little bit messy. And what we are getting, see here, the lab returns this result and is saying the antibodies got garbage, it's a lot of bands around, we can't use it. Look what have they been using. 30 minutes room temperature, 1% milk. If they just extended, that was what I recommended, extend the blocking time and increase to 5% milk, and the signal to noise ratio was much, much better. So for this antibody, incubation blocking at room temperature was working well. But we have other cases like this one. In this case, the blocking had to be done overnight. So as you see, that step depends upon your protein antibody pair. I can give you some guideline that if you work with proteins of high expression, and then probably blocking, if you don't load too much of a protein per well, then you probably don't need to block overnight. Here, just a reminder of a titration between the antibody and protein load per well, which will also clean the background, because we are after that. We are after signal to noise ratio. The signal for good antibodies, as you know, will show up within seconds. Membrane blocking and stripping and reprobing. I bet that many of you are probably doing the stripping. I'm not so fond of that procedure because you have to remember that you're breaking the bond between the primary antibody and the target, but at the same time, you may also remove some target from your membrane. So subsequent use of such membrane may result in more background. But also how you can take care of it is by, if you're stripping membranes, incubate them, block for longer time, and increase the blocker concentration. And comparing antibodies. This is very often done, and I'm not recommending it because you cannot compare an antibody to actin, which is high abundancy protein, to another antibody made to protein of low expression. It's uncomparable. The expression levels are different, and therefore you cannot directly compare such antibodies. Each antibody protein pair is unique. Here is an interesting case. I included it just out of curiosity for you. Most labs are using TBST buffer, but once the laboratory returned the result with PBST buffer, where the background was decreased. Why this has happened, I don't know. I just want to show you that when you're refining your blots, maybe changing the incubation buffer to PBST can help to clean the background. Please remember that PBS buffer is not suitable when you're working with phospho-specific antibodies. And now we are coming to the end of the Western blot protocol, and I would like to show you how to save time, which is very precious. And I wonder if any of you have ever done this. This is a parallel incubation of 3 primary antibodies at the same time on the same membrane. This result was submitted by one of the laboratories, and of course, I wouldn't recommend it for when you're using unknown antibodies. Here, you can do it when you know how the antibodies are performing. And also, the proteins which they are detecting should be apart by at least 20 kilodaltons, okay? But it works. So if you have some very precious set of samples, you can actually reuse use several antibodies on the same membrane at the same time. Another method to save time is to incubate everything at once. Standard protocol requires several hours of different incubations. Actually, for high abundancy and high medium abundancy proteins, you can do blocking primary and secondary at the same time. This will save you considerable amount of time. In this example, we are showing PSBA antibody. So this is an antibody to a very abundant photosynthetic protein. And a third method, of course, remember that this is recommended for antibodies which are already, you know how they are performing. I wouldn't do it for the antibody which I'm using first time. And a third method, directly conjugated primary antibodies. So the antibody is conjugated with enzyme or dye. If you're using custom-made primary antibodies, there are also kits which can, with very little hands-on, which will allow you to conjugate your primary antibody to an enzyme or fluorophore. And remember that this also shortens, of course, Western blot time, but is not recommended for transcription factors, because there we need an enhancing effect of secondary antibody. And now we are coming to secondary antibodies. They can be done in different hosts. You may feel sometimes a little bit lost because companies are offering a sea of different secondary antibodies. Just a reminder how the antibody looks like, and when you are reading in a product information sheet, it's written that a secondary antibody is made to whole molecule IgG, H and L. And H and L means heavy and light chains. Okay, so it means that the antibody, secondary antibody which you're going to use, is made to a whole IgG from rabbit, goat, mouse, or chicken. And just a reminder, antigen binding domain is the most important part of the antibody. And we have Fc region. Secondary antibodies. So if you have a primary made in rabbit, a host is rabbit, then you need anti-rabbit secondary antibody. And one would think that I take any secondary antibody, it just does not matter which one I'm taking and using. And it actually does matter because they can have different sensitivity and they can be used in different dilution. Usually people, when using and purchasing the antibodies, they concentrate how much milligrams is in a vial. What you should look at is the dilution you can use the antibody at. So, for example, there will be a difference if you purchase one vial, 1 milligram, and the antibody can perform up to 50,000. And if it goes only 1 to 5,000. So pay attention to when you are buying both primary and secondary antibodies, because if you can use the secondary antibody in high dilution, you will have lower cross reactions. And please do not incubate secondary antibodies longer than 1 hour at room temperature. If you are tempted for overnight incubations, I'm really, I think it will contribute to more background. That's what I saw. Once the lab submitted a result, they have been doing Western blot for 3 days using Rubisco antibody. And of course, as you may guess, the membrane was completely black. With Western blot, the shorter the better. Of course, there are exceptions. If you're working with more difficult proteins, then it may require longer incubations. And here, I just want to help you. I received the questions, which secondary antibodies to use for a normal Western blot on, for example, plant on algal samples. And which one would you choose? The one which is just simple one, goat anti-rabbit IgG having light chains, or the one which is also having minimum cross-reactivity to bovine, human, goat, mouse, and rat IgG. Think for yourself, which answer would you give? The correct antibody is the first one. Of course, you can use both, but usually the adsorbed secondary antibodies are more expensive and they are more suitable for immunolocalization. What it means, all this, it means that the secondary antibody has been passed through the columns with the IgG from this species and hopefully shouldn't have cross-reactivity to IgG from these species. But we are not working with goat mouse samples. So therefore, the simple secondary antibody is enough for our purposes. And now we are coming to detection, and probably most of you, or at least 60%, I bet, is using hemiluminescence. Hemiluminescence is still most popular detection method. We are using secondary or primary antibodies conjugated with an enzyme, horseradish peroxidase, and we require CCD camera to record the result. And here, this was one of the questions also I have received. Looking at the first blot, one would say, wow, so much background. No, this is really bad. But what happened here is that too sensitive ECL has been used on this high abundance protein. What you can do is you can just wash the membrane and develop with less sensitive ECL. If you win in today's quiz, you will receive both of them. So they are interchangeable, and depending upon what protein you aim to visualize, you can use either the one with super sensitivity or the one which is suitable for high and medium level of expression proteins. So advantages of ECL is sensitivity, allows quantification. Disadvantage is that recording has to be done promptly. It's suitable for quantification and analysis of proteins present in low levels. You probably, at certain point, if you work with ECL, you saw ghost bands. And the question is, why are they coming? It can be that you have too much of everything. You have too concentrated secondary antibody, too much protein loaded per well. It can also happen when you have antibody mismatch. So to get rid of ghost bands, you need to optimize your protocol and probably increase the dilutions of your antibodies and decrease the protein load per well. If the blot does not develop properly or something went wrong during documentation, can the ECL incubation be redone? I think you already know that answer, because the answer is yes, within the same day. So, for example, if you use too sensitive ECL and there is a lot of bands, you are aiming at such result, but it didn't come, you can wash the membrane and develop it with less sensitive ECL within the same day and save your experiment. Second most popular detection method is fluorescence. We are using secondary antibodies which are conjugated to fluorophores, and here you can see an example of simultaneous detection of two different proteins. So this method is suitable for multiplexing with several antibodies. It requires dedicated equipment, and it has sometimes lower sensitivity compared to chemiluminescence. It is in Western blot, there are no better or worse, really. Sometimes for some protein antibody pairs, fluorescence gives better results, and sometimes it will be ECL, chemiluminescent detection. It has happened for me that there were secondary antibodies provided with the equipment for fluorescent detection were not compatible with primary antibodies. So that was very puzzling. And by changing a secondary antibody, the protein was visualized using fluorescence. Fluorescence is suitable for quantification and multiplexing. And a third method is a colorimetric method. So we have an enzyme, alkaline phosphatase, and we do not need digital camera. The result can be recorded with the naked eye. The blot can be stored in a lab book, although it will fade over the time. It's inexpensive and fast, but not so sensitive. So if you do not visualize your target protein using this method, you should probably try chemiluminescence. I have seen such cases over the years. This method gives quick assessment, and keep in mind it is not suitable for proteins of low abundance. And no PBS containing buffers when you are using this colorimetric method. Hopefully, you are obtaining your results using one of those three detection methods, and they are looking satisfactory. How to determine molecular weight of a target protein detected on a Western blot? Why did I put that question? Well, I put that question because it has happened that people assigned the markers wrongly and the whole result was misinterpreted. We are using markers with IgG binding sites. They are compatible for chromogenic, chemiluminescent, and fluorescent detection. So if we're using ECL, then we are mixing the colored markers with the magic marks, and then we will have visualization during development of the Western blot. This is very important. Please keep this in mind to be sure where that you have the markers properly aligned on your blot. Magic marks have a very nice step in molecular weight. How to get an intense signal from Western blot using a weak antibody? That's also a very interesting question. And I would say that we need to start with theoretical analysis first. So sit at a desk and consider sample type. Have you used right organ? Was protein of low expression? Did you use fractionation? What was protein load per well? Maybe the load was too low. Did you have optimal transfer conditions? Was your protein maybe not transferred at all because you applied conditions which were not suitable for high molecular weight protein? Or maybe your target protein was blasted through the membrane and is available in the buffer. Did you use antigen purified antibodies and appropriate detection reagent? So for low abundance proteins, you cannot use alkaline phosphatase and colorimetric detection. So all this, all in all, is called optimization. And here the result, what can be obtained before and after optimization. I hope you are curious what have been changed. And if you check here, it is just a change in how much protein was loaded per well. Other parameters were kept the same. This also points, if you need to load only 2 microgram, you probably could shorten the whole protocol by incubating 1 hour at room temperature. The overnight incubations often contribute to increased background. I have advice for beginners. Record correct band first before multiplying variations of conditions. Sometimes researchers are sending me blots after 5, 10, 15 attempts. And I really think that the help can come after first 2 blots maybe are done. And you should go to the desk, sit down, and calmly reconsider, do the theoretical analysis as this presentation is showing you. Record the correct band first before multiplying variations of conditions, or reconsider your protocol. Here I'm showing the protocol which we are using for newly produced antibodies when we don't know their performance. So wild type and specific mutant is used. Protein load is high, as you see. Blocking is done 1 hour room temperature, but as you see here, primary antibody is very concentrated and is incubated overnight, and secondary antibody, high titer Agrisera antibody, only 1 hour room temperature. Of course, such protocol may generate a lot of background, but if we record the target band, we can afterwards optimize it. And by adjusting primary antibody concentration and protein load, we can get a cleaner result. So if you work with an unknown antibody, I would advise you to simply overload the whole system and then optimize it after you record the correct band. And please remember that you need to use validated antibodies or validate them yourself, so you're certain what they are detecting. And here I have an advice for experienced users. Look over the protocol which you use. The same protocol does not fit all antibodies. That's the most common mistake. So I hope that after optimization efforts, you are going to obtain much cleaner, much better results. And if you're still getting such blots, then you're most welcome to drop us a mail. I'm very eager to help. And also now I would like you to, we're doing a quiz, but you're going to answer for yourself. Common misconceptions about the antibodies. Other antibodies I've bought from Agrisera work, therefore this one should work too. Think for yourself, true or false? False. That one antibody works from certain company doesn't mean the others are going to work too. As you have learned, I hope today, it depends upon different factors. Antibody works for species A. For sure, it will also work for species B. Think your answer, true or false? False, because what determines if antibody is going to work is the sequence, which needs to be repeated in the protein you're aiming to detect, or repeated. It needs to be present in the protein you're aiming to detect. Same protocol can be applied for all antibodies. I think I can be pretty quick here because you already learned that in a previous slide. This is false. You always need to look at the properties of the protein you're aiming to detect. This is where it starts. And you need to match the proper and the correct antibody, not just hoping that it's going to work. Antibody gives a good signal in a Western blot, therefore it should also work in other techniques. True or false? Think for yourself. False. One technique, one validation. The antibody which is not giving good results in Western blot can still work quite well in immunohistochemistry. So please remember, one technique, one validation. You cannot assume that an antibody which is working very well in Western blot is going to perform well in immunoprecipitation. It all depends upon epitope exposure and antibody binding. And now I would like to ask you a question. Which information about antibodies should be included in scientific publications, and why is it important? I don't know if you are aware that half of antibodies used in research cannot be properly identified. It may be happen that you wanted to reproduce a result from your colleague, but antibody number and supplier was missing, or there was a supplier, but it was no number. If a company is providing 20 antibodies to GFP protein, it will be really difficult to know which specific one was used in that experiment. So please remember, for yourself and for those colleagues who are going to reproduce the experiments, include always antibody name, number, and supplier. Of course, Western blot protein details would be also good to have, as you maybe learned today, but at least number and supplier. And this is not an advertising for a supplier. It's a help to increase reproducibility of obtained results. I hope you're going to take it seriously. And I would like to also tell you that we have a technical blog where we are posting different issues, describing different results. And if you have blots you would like to share, I would be happy to also include them in our technical blog. If you have questions which you want me to address or solve them, please contact me, and I will be happy to include it in technical blog so others can also have access to it. We also have Western blot resource and interactive guide where you can find different tips regarding the Western blot. This presentation is like a living thing. I have conducted over 10 of them this year, and that one which I am doing for you today is much more developed compared to the one on the beginning of the year, because we are learning all the time. We are also having free resources. If you are interested, you can receive or download Agrisera educational posters. They are about photosynthesis, but also epigenetics. And the poster next year is going to be about protein extraction. I hope it's going to be useful. Please don't forget also that we have images from the posters, which are available for free use. And if you're curious about which conferences are coming next year, you can check it in the Global Plant Events calendar, which we are running for the 5th year next year, together with Plantae and American Society of Plant Biology. If you would like to receive Agrisera newsletter, you can notify that by email. And now a surprise for those who do not know, we offer over 100 free antibodies, and there's no limit on provided antibodies. They are done to proteins from plants, bacteria, and algae. And we would like to receive from you in return the results. We will provide those antibodies together with secondary antibodies and our ECL. The complete list is the link is here, and the list is updated each week. We are also welcoming antibody suggestions. If you would like us to develop the antibody, which could be helpful for you and for others, please let me know. Suggestions are always welcome. And now we are coming to the end. So for inquiries from England and Ireland, you're welcome to contact the host of our webinar, Dr. Magali Dale. And for inquiries from other countries, you're always welcome to contact support@agrisera.com. If you are curious which countries were present today, I can tell you that it was France, Portugal, Spain, Poland, Czech Republic, Japan, India, Canada, US. So there was also an entry from Argentina. So thank you very, very much for wanting to participate in the workshop today.
I think you've got a last minute question, Joanna.
Okay.
Is the hierarchy is to select the right tissue organ?
Is the hierarchy. It depends upon where protein is expressed the most. So you can use the databases to try to guide you. So, for example, if you know that the protein is present the most in senescent leaves, then you should use the senescent leaves. Or you should use, if it is pin protein, you should use the root tip. So if you do this theoretical analysis, it will really help you later to obtain better result. Do you have any idea why you find proteins in Western blot signals in higher kilodalton using MS? I didn't understand the last part. Can you rephrase the question?
Or just unmute if that's easier.
You are analyzing the band, I assume, and then but if you are analyzing a band using mass spec, you will always find not only your target in it. Why do I find a protein which is supposed to be smaller? Okay. Okay. Are gel gradients also important? Gel gradients are very good because they give a better separation based on molecular weight. So we are actually very fond of gradient gels. And you can custom yourself or you can purchase them. They will, especially if you, for example, if your protein has different isoforms, which differ by, let's say, a few kilodaltons, a gradient gel will be also helpful to distinguish between them. And Anne, I will address your question by email about your issue with signals in a higher kilodalton using MS. I would like to thank you one more time for joining. You were a wonderful and active group. And we have 36 responses in a quiz. Great, great. I promise it's going to be a lot of prizes. And if you have any questions in the future regarding antibodies and Western blot, please do not hesitate. You are always welcome to write. To contact Agrisera or Magali if you are based in the UK and Ireland.
And Joanna, I also wanted to thank you for this insightful workshop. And I think we truly appreciate your expertise and the time you dedicated to this session to provide really valuable insight on the Western blot technique. So thank you. And also, yeah, like you said, a big thank you to everyone for joining us today and for your active participation. I think it's nice to receive questions and I think your engagement have made this workshop really a success. So thank you very much. And like Joanna, I say, if you want, if you have any further question or if you like additional resources, yeah, please feel free to email us, and we're always happy to help.
Definitely, and it was the most international workshop which I have done this year. So thank you very, very much.
Yes, thank you very much, and thank you, Joanna.
Yep. Thank you, and also have a nice holiday break. And yeah, I hope we meet online or by email or on a conference in the future. So bye-bye.
Everyone, bye. Thank you. Thanks, Joanna. Bye.